Good day, everyone. My name is Ruel Vipatu, and I will be presenting my journal article analysis on analytical chemistry. My chosen journal is entitled PCR combined with lateral flow immunochromatographic assay to differentiate the narcotic Mitragyna speciosa from related species and detect it in forensic evidence. So the authors of that journal are Chayapol Pangpathong, Tatri Padongchoryon, Rongchai Suksawati, and Suchada Sokrong from the Chulalongkorn University of Thailand. Now, without further ado, let us now move on to the introduction. First, let us know what is Kratom, also known as Mitragyna speciosa. So basically, in Thailand, there are 4 different species of Mitragyna, the genus Mitragyna. And those are Mitragyna rotundifolia, Mitragyna diversifolia, Mitragyna hirsuta, and the last one being Mitragyna speciosa, which is the main focus of our discussion. Mitragyna speciosa or Kratom is peculiar from its 3 relative species because it is the only species of Mitragyna found in Thailand with psychoactive properties, namely mitragynine and 7-hydroxymitragynine, which is its derivative. M. speciosa or Kratom is used in ethnomedicine. It is used in relieving pain, inflammation, diarrhea, and etc. M. speciosa is also considered a narcotic plant and is illegalized in several countries like Thailand, Denmark, Bhutan, and many more. So why is it illegal? Its psychoactive properties has adverse effects to humans. So that is the main reason. And those adverse effects are muscle spasm, pain, difficulty in sleeping, watery eyes and nose, hot flushes, fever, and loss of appetite. In worst case scenario, kratom overdose can even cause death. In fact, 91 Americans died of overdose in which kratom was involved in. So what is the importance of distinguishing M. speciosa from its other relative species? The first reason is kratom has other relative species in Thailand, which are not illegal, which means it can be mistaken from those 3 other species, which can lead to a huge confusion to the authorities. And that leads us to the next reason. A rapid, cheap, and easy method to identify kratom is needed because with it, it'll be easy for the enforcers to identify suspected kratom samples that they'll get in the future. There are various methods for identifying kratom that had been reported. One of them is this one: combined technique of microscopic and chromatographic analysis, which has been proven to be an efficient method for confirming kratom identification. But the similarity of morphological characteristics of plants in the same genus causes complications in assays, which can lead to unreliable results. The next method is enzyme-linked immunosorbent assay, also known as ELISA. ELISA exhibits several advantages over chromatographic analysis as a result of its simplicity, selectivity, and sensitivity. It was a very promising method, but cross-reactivity of the anti-mitragynine antibody against the related compound was observed, which can also lead to false positive results and made the method faulty. Moreover, weather conditions and growth stage might also affect the said method. But consequently, DNA which is not affected by weather conditions or other aspects of the environment has been applied for plant identification. 4 core DNA barcode regions including ITS, MATK, RBCL, and PSBA-TRNH, intergenic spacer regions, have been used for the identification of plants. And based on DNA barcode sequences, polymerase chain reaction coupled with lateral flow immunochromatographic assay or PCR-LFA was developed. That concludes the introduction of the article. Now let us move on to the methods used in the study. So the first one is this one: plant materials and cases of suspected Kratoms. What they did here is an assigned taxonomist and co-author of this article, Associate Professor Tatri Padangcharyan of the Chulalongkorn University, first did the collections of sample. He collected 4 different species of Mitragyna, namely this one, M. speciosa in letter A, M. diversifolia in letter B, M. hirsuta in letter C, and M. rotundifolia in letter D. Now you can see the similarities of these plants with each other because they are from the same genus, which is the Mitragyna. The gathered samples were packed in a voucher and then sent to the Museum of Natural Medicines in Chulalongkorn University. 8 suspected kratom samples of different forms were received from unnamed officers from the southern part of Thailand. Procedure is the DNA extraction. They extracted 100 milligrams of each of the 4 species of Mitragyna through the DNEC plant mini kit. The DNEC plant mini kit is a commonly used kit in molecular biology laboratories for regular genomic DNA extraction from a variety of plant materials. All of these suspected kratom samples were subjected to genomic DNA extraction using the said kit. They modified the procedure by adding centrifugation step at 16,000 RPM for 20 minutes prior to DNA extraction. That they begin the next step, which is the selection of the unique DNA regions of M. speciosa for primer design and duplex PCR amplification. They're going to use that for the identification of M. speciosa from its other relative species. And they proceeded to employ single-use PCR for visual detection of labeled PCR amplicons. Polymerase chain reaction coupled with lateral flow immunochromatographic assay or PCR-LFA is a visual diagnostic kit with a nylon-based strip used to detect DNA amplicons. The polymerase chain reaction or the PCR, often known as the molecular photocopying, is a quick and low-cost method for amplifying or copying tiny portions of DNA. Since molecular and genetic investigations require considerable volumes of DNA, examinations of isolated pieces of DNA are almost impossible without PCR and PCR amplifications. Meanwhile, the LFA technique includes the identification and suspension of antibodies and antigens. Using particular antibodies on lateral flow strips, marked PCR amplimers are suspended and then coloring agents display a color signal on the strip. LFA is a rapid, cheap, portable, and easy-to-use test in identifying infectious disease, narcotics, and the most common one is pregnancy. Basically, LFAs look like a cassette in which you put a sample on the sample well. The sample well is the circular hole on that cassette, and then it will show colored lines with different results. So the PCR LFA system that they use here in the study was manipulated for visual detection of duplex PCR products amplified from Mitragyna plants. This is a 2D illustration of the cassette that they use. So there are 3 lines there. So the first line is the internal control line. The second line is the MS-specific line. And the third line is the strip control line. The last part of their methodology is the specificity, sensitivity, and rapidity of the test. So to confirm the specificity and assure the absence of cross-reactivity, They subjected DNA from 32 authentic Mitragyna samples collected from various parts of Thailand to PCR-LFA to ensure the specificity and ensure that there is no cross-reactivity. And that concludes the methods and material part of the article. Now let's go to the results of the study. The results of their study ended up a success. Out of 4 core DNA barcode regions, they tested for the selection of species-specific primer. Out of 4 core DNA barcode regions, they tested for the selection of species-specific primer, namely MATK, ITS, PRNH, PSBA, and RBCL. They found out that MATK is the fittest DNA region for the discrimination of Mitragyna speciosa from the rest of the species. It was chosen for the reason that it contains species-specific nucleotide indels exclusively found in M. speciosa, which means they can use that in the differentiation of M. speciosa from the other species because it is only found in M. speciosa. It was then used as the M-specific primer. The PCR-LFA done in the study showed positive results. Regardless of the location origin of the samples, all of them showed 3 black lines, which imply that the assay has a high specificity for M. speciosa. For other closely related Mitragyna species, only the internal control primer applicon was immobilized on DNA immunochromatographic strip, meaning they didn't have 3 black lines. So this indicates that PCR-LFA is a viable forensic diagnostic technique for the differentiation of M. speciosa from the rest of the Mitragyna species. Moreover, they also found out that dried, processed, and mixed samples exhibits gray color in the detection lines as opposed to black. This outcome is possibly caused by the low amount of M. speciosa DNA in them. Now let's move on to the authentication of suspected forensic samples. So these are the results. All the forensic samples received from the anonymous officers in the southern part of Thailand. As you can see, all of them has 3 black lines, except the sample KE04, which only has 2 black lines. It does not contain M. speciosa DNA. Also, you can see that some of the lines are not that black. They are more of a grayish color. So this outcome is possibly caused by the low amount of M. speciosa DNA in them. And that concludes the result of the study. Let us now move on to the discussion. So these are the main discussed points in the article. The first one is the need for a simple method in Kratom investigations. So as mentioned earlier, Kratom is a narcotic plant. And due to the risk of drug abuse of M. speciosa or Kratom, several research groups have attempted to develop simple methods for Kratom investigations. Recently, the competitive ELISA and the other one that we mentioned earlier with the long title have been discovered to have faulty results, which means they can produce false positive results because of cross-reactivity. Therefore, genetic testing by using DNA barcode sequences was applied and developed as a simple method for the identification of M. speciosa. Also mentioned earlier, 4 core DNA barcodes were tested and they ended up using the MATK, which leads us to the next discussed point, this one, why MATK was chosen for M specific primer. Then the primer-dimer hybridizations from duplex primers were checked with the multiple primer analyzer program to avoid primer-dimer formation. The discussion of the processing of samples and their results. So then the researchers concluded that the results showed that the PCR LFA system is a fast, reliable, and economical tool for the detection of M. speciosa in forensic analysis, which is the analysis. Now, my personal conclusion about this, I believe that this article is a strong scientific article where they have established the purpose of their study very well by stating the problem with the M. speciosa being illegal and harmful to humans. which then led to them introducing the primary focus of their study, which is the usage of PCR LFA. Furthermore, they have stated all of the procedures that they have done in their article in a very detailed way. They have also clearly and neatly compiled their results from all of the procedures that they did. On the other hand, I don't think that this article's target readers are the general public because this article is overflowing with technical and scientific terminologies that an average reader like me would not possibly understand. Now, in terms of being a scientific study, it is also a strong one because it has successfully achieved its goal or purpose. Now, the possible limitation that I can see in their study is this one. PCR LFA is limited to the detection of the substance and it does not determine the amount of M speciosa DNA in the sample. And that is all. Thank you for listening and I hope you learned something.